Hi everyone, this will be learning outcome number 6 for module 10, and we're going to be covering the synapse. A synapse is the site of communication between either two neurons or between a neuron and an effector cell. This effector cell can be either a muscle or a gland. And synapses are going to be essential for homeostasis just because they will allow the information to be filtered and integrated into the rest of the body to cause a response. And then synapses are also going to be important because some diseases and neurological disorders will result from the disruptions of these synaptic communications. And there are going to be many therapeutic and addictive chemicals that will affect the body at these junctions. So that's why it's important for you to understand the mechanisms that occur between these communications. Therefore, on these images, we can clearly see a synapse of a neuron with another neuron on the first image. So this would be the cell body with the dendrites and the axon. At the end, you're going to have the terminal buttons, which will then synapse with either the cell body or the dendrites of another neuron. In the second example, we have the synapse of a neuron with a skeletal muscle, and we already talked about this, how this will create what we call the neuromuscular junction. And the third one is what we call the neuroglandular synapse, which, like the name says, it's going to be a synapse of a neuron with glands. So let's see how these work in the next few slides. First, there are some terms that I think that's important for us to cover before we move on. Therefore, a synapse between two neurons, the neuron that's sending the signal is what we call the presynaptic neuron. That means a neuron prior to the synapse. And the neuron that's receiving the message is called the postsynaptic neuron. And we can see over here on this image, this one that's sending, for example, the signal is the presynaptic neuron, sending to this other neuron over here. So this one would be your postsynaptic neuron. And you can see how this postsynaptic neuron is receiving input from this other presynaptic neuron as well, right over here at the cell body and at the dendrites. Then we have another neuron that will be synapsing down here at the terminal buttons. Therefore, we can say that these neural synapses, they're going to be named based on the parts of the neurons that are going to form the synapse and also the direction of the information flow. Examples of these include the axodendritic, as we can see over here. So it's a synapse from the axon to the dendrite. Then we have what we call the axosomatic synapse, which will be from the axon to the cell body, like the name says. And the last one is going to be called the axoaxonic. So definitely from the axon to an axon, as we can see over here. But all of them will start with an axon because that's where the information that's traveling through that presynaptic neuron will end to be able to pass on to the next neuron. And most neurosynapses are either axodendritic or axosomatic. Very few are going to be axoaxonic. Basically, we have two different types of synapse. We have what we call the electrical synapse and the chemical synapse. The electrical synapse is going to be your action potential, which we talked about in the previous learning outcome. So it's going to involve channels that will allow ions to flow from one cell to another. And the chemical synapse will involve neurotransmitters or hormones that will be released into a space between the presynaptic neuron and the postsynaptic neuron or the effector organ. And we do have about 100 different types of substances that can be classified as neurotransmitters. Here we have some that are listed. And these are either going to be excitatory or inhibitory. And we will talk about what does it mean to be excitatory or inhibitory in the next few slides. First, just to review a little bit what an action potential is, here we have this area right over here, which is called the axon hillock or also the trigger zone. So remember, this is the point of no return. So think about this will be your postsynaptic neuron, for example. You're going to have other neurons that are going to be synapsing either with the dendrites or the cell body of this postsynaptic neuron. And once you have enough of these synapses occurring here, it will create an action potential that will start right over here at the trigger zone and move down the axon until it reaches the terminal buttons down here. So how will this action potential occur? Well, basically, it will start with sodium coming in to the cell and potassium going out of the cell. So once you have sodium going in and potassium going out, it will create a depolarization of this membrane that will then transmit the signal all the way down the axon to the terminal buttons. Analyzing this image in a little bit more detail, we're going to see that what's represented here in purple is going to be the resting membrane potential. So this will be at around minus 70 millivolts. And the action potential is starting right over here at the trigger zone. Once sodium enters, then you're going to have the action potential moving from this area to the next area of the membrane and depolarizing the next area. And you can see how the area of the resting membrane gets shorter and shorter as we move along. And the area where the sodium is depolarizing the membrane is moving along towards the terminal button. And at the same time, you're going to have the potassium going out of the cell. And when the potassium goes out of the cell, it repolarizes the membrane. And then at the end, you're going to have an area where you're going to have the repolarization of the membrane after it's become hyperpolarized due to the action potential. So it's a process that goes down the axon all the way to the terminal button, and this occurs due to the exchange of sodium ions entering and potassium ions exiting the cell. There are two factors that influence the velocity of the conduction. The first one will be the diameter of the axon, and the second one is the presence or the absence of myelin. Now, the larger the diameter of the axon, the faster the action potential is going to be conducted. And this has to do with the fact that the axon with a larger diameter, it will offer less resistance to the local current flow, which allows adjacent regions of membrane to be brought to threshold more quickly. So therefore, the thicker it is, the faster it will travel. With regards to the presence or the absence of myelin, the conduction of action potentials is going to be more rapid along this myelinated axon than along an unmyelinated axon. And this has to do with how the exchange of ions occur in a membrane that's myelinated and a membrane that's unmyelinated. So in a membrane that's unmyelinated, like we see over here, we're going to have what we call a continuous conduction. Therefore, for the action potential to travel, the membrane will have to depolarize along the whole length of the axon. So there's going to be sodium going into the cell and potassium coming out of the cell, but it has to travel along the whole length of the axon. And on a myelinated axon, the conduction is what we call saltatory, which means that the exchange of ions will only occur in the spaces between the myelin, and these spaces are called the nodes of Ranvier. So that's why it's called saltatory, because it jumps from this area over here, which is the trigger zone or the axon hillock, to the next area of the node of Ranvier. So the area where the myelin is, it does not have to be depolarized, and therefore it jumps from this area to this area, and then to the next area where the node of Ranvier is, and then to the following one. So this is why it's faster, because it doesn't have to depolarize the whole axon, only certain areas of the axon. Okay, now that we revised the electrical synapse, we can talk about the chemical synapse, which is what will occur at the end of the axon, more specifically at what we call the axon terminal or the terminal button. So here coming down, we're going to have the axon hillock or the trigger zone, and therefore the action potential is coming down over here, jumping from one node of MVA to the other once it reaches this area right over here. And what's happening on this terminal end is the same thing that's happening right over here, right? So the action potential came down the axon all the way to the axon terminal, and now it's synapsing with either the dendrite or the cell body. So this axon over here belongs to the presynaptic neuron, and this neuron over here will be your postsynaptic neuron. So the action potential comes down over here. Once it comes down, what happens is that you're going to have an influx of calcium. So calcium is going to come in through these channels over here that are going to open up. And once there's that influx of calcium coming in through the channels, this will cause the synaptic vesicles that contain neurotransmitters to migrate down towards this space that's called the synaptic cleft, and it's going to release the contents, which are again the neurotransmitters, through exocytosis. So notice how the membrane of the synaptic vesicle will fuse with the membrane of the presynaptic neuron and therefore release these neurotransmitters. And these neurotransmitters, they will bind to these ligand-gated channels. And if these neurotransmitters are excitatory, then what will happen? They will open up these ligand-gated channels and sodium will come in through them. So sodium comes in, but what binds to the ligand-gated channels are the neurotransmitters. And if you have enough sodium coming in, then it will start to depolarize this membrane. So if you have, again, enough sodium coming in on this postsynaptic neuron over here, it will reach the trigger zone right over here, and then it will cause the depolarization of the next neuron or the postsynaptic neuron. So this is how the synapse occurs. Now, a neurotransmitter is either going to have an excitatory or an inhibitory effect on the membrane of the postsynaptic cell. So if the binding of a neurotransmitter to a neurotransmitter receptor opens or closes the ion channels in the postsynaptic membrane, the neurotransmitter is either going to depolarize or hyperpolarize the postsynaptic cell. Therefore, a neurotransmitter that depolarizes the postsynaptic membrane is said to be excitatory because it will bring the membrane closer to threshold to be able to cause an effect or the depolarization of the membrane. Therefore, a depolarizing postsynaptic potential is called an excitatory postsynaptic potential or EPSP. And although a single EPSP normally will not initiate an action potential, the postsynaptic cell does become more excitable. But because it is partially depolarized, it is more likely to reach threshold when the next EPSP occurs. So the more EPSP that occurs, the more the cell will become excitable to be able to cause an action potential and depolarize. In contrast, if you have a neurotransmitter that causes hyperpolarization of the postsynaptic membrane, it will then be inhibitory. So during hyperpolarization, the generation of the action potential is going to be more difficult than usually because the membrane potential becomes inside more negative and therefore even farther from threshold than it is at the resting state. So a hyperpolarizing postsynaptic potential is going to be called an inhibitory postsynaptic potential or IPSP. Now, common examples of excitatory neurotransmitters are acetylcholine and glutamate, for example, and Common examples of inhibitory neurotransmitters can be glycine and GABA, but some will actually cause both, sometimes excitatory, sometimes inhibitory, and this will depend on the specific receptor and the location. And some examples of neurotransmitters that can cause both excitatory and inhibitory responses can be dopamine, serotonin, and norepinephrine. So here we have what we call the summation, or adding on all the inhibitory and excitatory responses. So if you notice over here, we're going to have more excitatory because we have 3 excitatory responses from these neurons that are coming here, and we have 2 inhibitory responses. Therefore, we have more excitatory than inhibitory, and therefore it will cause probably an action potential at the trigger zone. And this will be, in this case, it will be an excitatory one, so it will travel down the axon. So it all depends on the net summation of the excitatory and the inhibitory depolarization to see if it will reach threshold or not to cause this action potential. And I like this image here because this image is showing you how many terminal buttons are synapsing with this cell body and with the dendrites of this cell body. So the terminal buttons are the tiny blue structures over here, and this would be the axon of this neuron where the cell body is over here, and these will be the dendrites of this neuron. So notice how we have all these terminal buttons synapsing with this one neuron, and all of them are telling this neuron, it's me, it's me, it's me. I am the information that's more important to be sent down the axon, passing through the axon hillock, other terminal button, and going down the axon. So this is what we call reaching threshold. So the more important the information is, the better chance it has of reaching threshold and propagating down the axon. All these small stimuli that are occurring at any given cell body or dendrite are what we call graded potentials. So here we have examples of graded potentials, which are these small, tiny spikes of information. You can have some that last a little time, others that last longer. Notice how this plateau over here is longer according to the time, and some are more potent than others because this stimulus over here is actually reaching threshold. So it goes from minus 70 millivolts, which is the resting membrane potential. All these graded potentials did not reach threshold, so they're over here at minus 60 millivolts. But one single one that was added reached threshold, went above minus 55 millivolts, and therefore it caused an action potential. Now you can have other graded potentials that will do the opposite, so they will cause hyperpolarization. So it all depends on how many of that same stimulus you're getting and at what frequency you're getting this stimulus. And this will result either on an action potential to be excited and propagate, or it will result in an inhibitory response where that information stops at that neuron. So here we have an example of an excitatory response and an inhibitory response. Excitatory in green, inhibitory in pink. Notice how the excitatory sort of wins the inhibitory, and at the end you have an action potential. Or the action potential can actually be hyperpolarized and be below minus 70 millivolts. Now the summation, so the addition of all these stimuli will cause 2 different types of summation, which are spatial and temporal summation. Spatial has to do with having several weak signals from different locations. So think about in a space, you're going to have several different weak signals that will come together and be converted into one large signal that will then propagate. And temporal summation has to do with time. So you're going to have several rapid, weak signals that are coming from the same source. So the same type of information over time. And because it is telling the postsynaptic neuron this information is very important over time, therefore this information needs to be processed. So that's what we call temporal summation. And here I did a little summary for you guys of the difference between graded potentials and action potentials so that you can compare the two.